thank Kathy Seibold for being here to support our webinar and introduce our presenters. Kathy, I'm going to turn the webinar over to you so you can introduce the topic and our presenters.
Thank you, Sean. Welcome, everyone, and thanks for joining in today. It is my pleasure to introduce Dr. David Myrold and Jordy Kersey. Dave Myrold is a professor in the Crop and Soil Science Department at Oregon State University. He is also involved in the Dynamic Soil Properties Focus team. Jordy Kersey is a graduate student at Oregon State University who has just completed her master's thesis under the direction of Dave Myrold. The research project they will be discussing today was funded by NRCS as a CESU grant agreement. The research objective was to identify soil biological properties that change in response to either management or natural disturbances common in forest ecosystems. I will now turn it over to Dave Myrold, who will kick off the presentation.
Thank you, Kathy. It's great to be with you this morning. Looking through the list of participants, hello to several friends and colleagues out there who I see signed up, and also to many of you who haven't had the opportunity to meet before. As Kathy mentioned, this project formed the basis of Jordy's master's project while she was at Oregon State University, and I am going to let her make the presentation. I'm here mainly for moral support, and then if there are some questions and answers afterwards, I can help out with those as well. Jordy did a great job, and actually a lot of the last data was collected when she had some permission to go into the lab and finish some things up, as we've been under COVID restrictions. And so under some unusual circumstances, she was able to finish things up, and I hope you enjoy her presentation today. Jordy, it's yours.
Great, thanks. Hi, everyone. Thanks for tuning in. Today, I'm going to discuss soil health and the response of the soil microbial community to disturbances across Western North America. Timber forests across the Northern Hemisphere are a vital component of the biosphere. Forest ecosystems mediate nutrient flow, are hotspots for biodiversity, and provide feedback on the climate system. Therefore, our understanding of the function of forested land is important to maintain and develop sustainable management plans which avoid accelerating the degradation of these systems. Across much of the western U.S. and Canada, large areas of both public and private land are managed, and many are dedicated to the production of timber. To ensure the continued productivity of these areas, we must appropriately manage forest stands to maintain sustainability. There is evidence that adequate silvicultural management practices require an understanding of forest ecosystem ecology. Therefore, we need to understand processes carried out in forests, both managed and unmanaged, to ensure ecosystem services that meet the long-term needs of a healthy stand are being provided. Forest ecosystem services include transformation and decomposition of organic material, nutrient cycling, water regulation, and physical substrate for above-ground vegetation. These services are provided by the soil and its inhabitants. Soil of high quality, which can perform these necessary ecosystem services, leads to highly productive stands and overall environmental and economic sustainability. In this way, high-quality soil sustains the desired vegetation and provides resilience to potential forest disturbances, and may be referred to as healthy soil. The concept of forest soil health implies assessing soil properties and processes as they relate to the ability of a soil to perform its necessary functions. It also places emphasis on soil biodiversity and ecological functions that make a soil a dynamic living resource. Soil health may be defined differently depending on the goals and the environment of the site of interest. For example, a managed timber production stand may determine soil health based on its ability to produce marketable biomass, while the soil health of an unharvested forest stand may be determined by its ability to maintain a diverse and complex ecosystem. Monitoring of soil and long-term sustainability of forest ecosystems relies on the use of indicators. In the case of soil health, an indicator would be a measurable soil attribute that helps to determine how well a soil functions. Soil indicators may be used to track relatively static soil properties, such as soil texture, structure, nutrient pool status, and pH, or they may also be implemented to track more dynamic properties, such as mineralization and nitrification rates, extracellular enzymatic activity, and moisture dynamics. Using a suite of soil health indicators provides a holistic view of the soil system as well as its functional capacity. Many dynamic soil properties important to maintaining ecosystem function are mediated by soil microbes. Finding indicators of how well the soil biological community is performing, as well as how its structure and composition might be developing over time, could be useful in determining the trajectory of soil health within a stand. This is particularly important subsequent to disturbance events, where soil biota may aid in ecosystem recovery and resilience to degradation. Based on the importance of understanding how forest soils respond to and recover from disturbance, this research was guided by two main objectives. The first was to determine the current state of knowledge about how soil microbial community dynamics change in response to forest disturbances. And second, to measure soil properties related to soil nutrient pools and microbial activity-related processes to investigate the impacts of harvesting on these soil health metrics. We addressed Objective 1 by an extensive review of the literature regarding microbial community response to an array of forest disturbances in an attempt to synthesize common trends and or gaps that may exist in the current literature. Objective 2 was addressed by an experimental study investigating harvesting impacts, specifically those related to organic matter removal and compaction, and how they affect soil health parameters that are often related to the soil microbial community. The impacts of disturbance on soil microbial community dynamics in Western North American forests, a literature review, aimed to address the first objective of this research. Forests have evolved to depend on patterns of disturbance for the succession and growth of the entire ecosystem. This is the case in both natural and managed forests. Although in many instances disturbances hold the same purpose within a forest, the actual event can vary widely in ecosystem response through the interaction of certain disturbance attributes, including spatial extent, severity, and duration. Disturbance types may differ spatially, with an area affected ranging from just 1 square meter to hundreds of square kilometers. They may differ temporally, with durations from days to decades, or they may differ in severity, from low to high impact. The severity of impact may be defined by several parameters, including homogeneity within the spatial extent, the inflicted level of vegetation stress and/or mortality, removal of the forest floor, changes in stand conditions, such as those related to temperature and moisture, and physical or chemical changes to the soil environment. These are likely to impact the response to the soil microbial community and ultimately the recovery of the ecosystem. For example, if we focus on clear-cut harvesting disturbance, which is represented here by the gray ellipse highlighted, this has a spatial extent ranging from about 10 to 50 square kilometers, a duration ranging from about a day to a month, and a severity of low to medium, depending on the harvesting technique that's implemented. Understanding where disturbances fall on this spectrum may enable better understanding of the potential impact and how the forest ecosystem may respond. The literature review summarized findings based on the published literature regarding the observed impacts to the soil microbial community in response to forest disturbance. We looked at disturbances of natural origin, including wildfire, insect infestation, and drought, as well as those of anthropogenic origin, such as timber harvest or restoration treatments, which included thinning and prescribed burning. Often restoration treatments are implemented to lower the risk of a more severe disturbance and may be considered as a preventative measure. We focus on the impacts of disturbance on three specific categories related to the soil microbial community. The first being the soil environment, which encompasses physical and chemical soil property changes. Then we looked at the soil microbial community itself by looking at abundance and biomass data, community composition data, as well as alpha and beta diversity metrics. And lastly, we looked at the response of soil microbial functions, such as those related to decomposition, symbiotic fungi, enzymatic activity, and nitrogen cycling. So let's take a look at some of the impacts on specific disturbances on some of these microbial-related metrics. Wildfire was found to negatively alter the soil environment, such as through the intuitive loss of organic matter, which supplies the microbial community with carbon and nutrients, as well as through the alteration of water dynamics, which may also constrain normal microbial function. Additionally, due to the inherent biological sensitivity to direct heating, fire had a strong negative effect on most microbial metrics, including microbial biomass, abundance, richness, evenness, and diversity. Fire also negatively impacts microbial functions by reducing the potential for heterotrophic decomposition, altering soil nitrogen dynamics, and degrading the mycorrhizal fungal community. Overall, these effects may be short or long-term, depending on the fire severity, as well as the extent of the area affected. Insect infestation impacts are distinct from other forest disturbances, largely because the temporal dynamic of an insect infestation is unique. Impacts from infestation may last for multiple years and create heterogeneous environments as trees are infected and go through succession at different times following an outbreak. Altered environmental parameters associated with insect infestation include reduced canopy cover, increased soil moisture, decreased fine root biomass, altered sea and in cycling, and changes to the water supply. All of these dynamics may have important implications for the soil microbial community and may significantly alter the composition and function. Overall, the reported impacts of infestation on soil microbial abundance and composition have been found to be variable. However, there is some evidence that lower proportions of surrounding tree mortality may mediate beetle impacts on the soil biological community. This is similar to wildfire, where severity and extent of damage largely determines the extent of negative impact on the soil community and related processes. After examining the reported impacts of natural disturbances on the forest soil community, it is important to look at the potential interactions which may exist between common natural disturbances and changing environmental conditions. As disturbance type and stand conditions shift in response to a changing climate, it is critical that we develop an understanding of how above and below ground biological communities will respond to novel combinations of disturbances. Northern temperate coniferous forests are already and will continue to be under pressures brought on by global climate change. These pressures include increased temperatures and altered precipitation regimes, ultimately leading to the expansion of areas impacted by drought-like conditions. Drought-like conditions may subsequently aid in expanding the range of common forest insect and pathogen invaders and also increasing above-ground vegetation stress. Such interactive disturbances may prime a forest stand to ultimately become more susceptible to natural disturbances, including wildfire and insect and pathogen attack. Combinations of disturbances which interact with one another inherently decrease forest resiliency, and this can create a positive feedback loop of disturbance, which leads to more disturbance, ultimately increasing the frequency in which a system is exposed to disruption. This is likely to increase the degradation of function over time. As I mentioned, timber harvesting is a widespread occurrence across much of the western U.S. and Canada, with thousands of acres of forest in some stage of timber rotation. It is important that we understand the implications of harvest disturbance in order to maintain the productivity of these areas for many years to come. In the aftermath of clear-cut harvesting, the soil surface is significantly altered. In some cases, the forest floor is removed, creating an environment with greater exposure and increased vulnerability. Even in areas where a portion of the residual organic matter is retained, the soil surface is still significantly altered by removal of vegetation. There is also the potential for heavy machinery-induced stress on the soil biological community through damage of soil structure and compaction. Typically, across all forest types and biomes, harvest is generally associated with a reduction in microbial biomass. And studies have shown the microbial community of harvested stands to be distinct from unharvested stands. This may indicate changes to the provision of ecosystem services depending upon how certain populations of microbial groups are altered within these community shifts. And in terms of microbial functions, timber harvesting has been shown to significantly alter C and N cycling within forest soil. This may have important implications not only for soil microbial community, but also for the regeneration and success of new species. As with wildfire and insect infestation, the extent and the severity of disturbance largely determines how long the system will take to recover. In contrast to severe wildfire, insect infestation, and timber harvesting, restoration treatments have shown to have potential benefits. Often, both thinning and low-severity prescribed burning treatments benefit the soil and overall stand environment by increasing nutrient availability, tree health, as well as pest defense. An unripe microbial mortality caused by direct heating during wildfire, often microbial biomass is reported to be unaffected by low-severity, well-distributed prescribed burning. Additionally, often microbial measurements made after thinning are undifferentiated from untreated areas. These lessened or positive impacts are an indication that low-severity disturbance implemented to reduce the risk of high-severity disturbance may aid in preserving these forested ecosystems and allow for increased forest resiliency. Overall, we found that non-restorative disturbance has a negative effect on soil bacteria and fungi, although the degree to which this is true varies by disturbance type. Often we see microbial recovery within 5 to 10 years, indicating the resiliency of the soil biological community. In general, the greater the physical soil disturbance, the greater the impact of disturbance to the microbial community. Additionally, disturbances with relatively low severity and in which impacts are widely distributed are found to have less impact on the soil environment and in turn the biological community. In addition to summarizing the impacts of disturbance on the soil microbial community, we were also able to detect areas where gaps exist in the literature. There are many studies which investigate the immediate effects of disturbance, but long-term longitudinal studies are less common and a potential aim for future research. Often studies investigate just one component of the complex microbial community, such as just looking at community composition or just measuring microbial biomass. Future research, which takes a more comprehensive approach by combining community composition data with activity indicators and soil health metrics, may be able to provide a more holistic understanding to how disturbance impacts the soil community. And now, before we move on to the second part, there is a poll question. How soil microbial communities rebound within 1 to 5 years, 5 to 10 years, 10 to 15 years, or greater than 15 years?
So everybody
take a crack at the poll question we have here, and then we'll share the results. All right, Jordy, I think the numbers have settled down, so I'm going to end the poll and broadcast the results if you want to talk about the responses.
Okay. So it looks like we have 38.6% said 1 to 5 years, 27.2% said 5 to 10 years, 25% said 10 to 15 years, and 9% said greater than 15. The correct answer is 5 to 10 years, but obviously this can be very variable depending upon the severity of the disturbance and other such properties that are individual to the stand. So now let's move on to the portion of research. The responses of soil health indicators to organic matter removal and compaction manipulation at 6 LTSP sites in the western U.S. As I mentioned, long-term studies are less prevalent in the published research than studies investigating the immediate impacts of disturbance in the community. In our experimental portion of this research, we wanted to investigate how soil health, especially properties generally related to the health and activity of the soil microbial community, would be impacted by a specific and widespread disturbance after at least 15 years. The Long-Term Soil Productivity Study, or LTSP, was initiated by the U.S. Forest Service in 1989 and aimed to address the impact of post-soil disturbance on soil productivity. The LTSP is a good example of a long-term experiment initiated not only to track the immediate effects of a pulse disturbance, but also the effects over the long term, encompassing dynamics throughout the entire rotation. The original site establishment took place in 1994 with 4 sites across 4 regions of the U.S. By including this range of ecozones in the initial experiment establishment, the design enabled investigation of how different forest ecosystems may have unique responses to harvesting disturbance. The main principle of the LTSP study is that within the constraints of climate, a site's potential primary productivity is regulated by physical, chemical, and biotic soil processes that are readily affected by management. Treatments were installed to investigate the impacts of harvesting treatments, specifically organic matter removal and compaction. Since its initiation, the LTSP experiment has expanded to over 100 core and affiliated sites across both the U.S. and Canada, making it one of the largest networks dedicated to forest ecosystem research. All of the sites that we sampled for our study were part of the LTSP network, either core or affiliated experiments. 3 of our study sites were core LTSP experiments and 3 were affiliated. The core sites are Council, California, and Umpqua, which are located in Idaho, California, and Oregon. And 3 of the affiliated sites are Matlock, Moala, and Fall River, located in Washington and Oregon. The California site is somewhat unique in that it consists of 3 individual sites, each with 1 treatment replication. The sites are within 20 miles of each other, and so we aggregated them to make a single California site with 3 treatment replications so that it would match the number of replications from other core LTSP sites. The LTSP sites we sampled had unique site characteristics including soil texture, the age sense harvesting, and dominating tree species. Treatments had been established between 16 and 25 years prior to our sampling. The core sites are the oldest sites included in this study, with Council being established 25 years prior to sampling in 1994, and California and Umpqua being established 22 years prior to sampling in 1997. The affiliated sites are a few years younger, with Fall River having been established 20 years prior to sampling in 1999, and Mawara and Matlack established 16 years prior to sampling in 2003. Despite Fall River not being the oldest site, it is the only site that we sampled which had reached canopy closure stage. This might be an indication of the productive capacity of this site being higher relative to the other locations. The core LTSP sites have a common, fully factorial experimental design consisting of 3 levels of organic matter removal. OM0 is bowl-only harvest, OM1 is whole tree harvest, and OM2 is whole tree harvest with removal of the forest floor. There are also 3 levels of compaction, C0, which is low compaction, C1, which is medium compaction, and C2, which is high compaction. For our study, we sampled 3 plot replications of all 3 levels of organic matter removal and low and high levels of compaction. In addition to the treatment plots at each site, we also sampled a nearby uncut stand to serve as our reference condition. The affiliated sites include part of the core factorial design. We sampled 4 plot replications of 2 or 3 levels of organic matter removal, all with a low level of compaction. At the affiliated sites that we sampled, OM0 is still bowl-only removal, OM1 is whole tree removal, and OM1 plus is whole tree removal with the addition of the removal of coarse woody debris. Similarly to the core sites, in addition to our treatment plots, we also sampled an uncut stand, which could be used as a reference baseline. For this project, we formulated 3 hypotheses. The first being related to previous findings establishing that low-severity disturbances, even immediately following the event, are unlikely to cause severe disruptions to the soil community. Therefore, after greater than 15 years, we hypothesized that only in the most severe organic matter removal plots, which would be OM2, would a decline in soil nutrient pools and microbial activity-related processes still be observable. Our second hypothesis was based on previous findings reporting a lack of impact from compaction treatments on other forest ecosystem attributes. Therefore, we hypothesized that compaction treatments would have less detrimental impacts than organic matter removal treatments after greater than 15 years post-harvest. And lastly, our third hypothesis was based on the inherent heterogeneity of forest soils and the individual site characteristics which exist at our study locations, such as differing soil textures, dominating tree species, and climate regimes. This led us to hypothesize that site differences may preclude a universal response of harvesting treatments and cause differentiated responses of some soil measurements. Our soil sampling took place in late June at Umpqua, Mawara, Matlack, and Fall River, and mid-July at Council in California of 2019. The sampling times coincided with maximum biological activity and represented the long-term response to harvesting, with harvest ranging from 16 to 25 years prior. Mineral soils were collected by shovel, taking soil from 0 to 20 centimeters depth at 5 points within each plot and pooling them to create 1 composite sample per plot. After collection, samples were kept on ice until returning to the lab, where each sample was sieved to 4 millimeters and air-dried for further analyses. For our laboratory analyses, we were particularly interested in 2 groups of soil property measurements, the first of which were nutrient pools. which include total carbon, total nitrogen, and active carbon. Total C and N are important because they represent the amount of these nutrients being stored in the soil, and in the case of C, the pool from which microbial energy may be obtained. They can also provide insight into potential nutrient limitations within the specific site or treatment. Active carbon has been suggested to be a measure of more labile forms of C in the soil, and has also been reported to be correlated to microbial biomass as well as total carbon. By measuring these nutrient pool properties, we could gather a sense for how the system had recovered in the greater than 15 years since the harvest disturbance. Often, immediately following harvest, these pools are reported to be degraded. However, this state tends to only last 5 or so years. By including these measurements in our study, we could get a sense for how the nutrient pool at these sites responded long-term to post-harvest disturbance. The second category of soil properties we were interested in were microbial activity indicators, such as carbon mineralization, net nitrogen mineralization, and enzyme potential activity. Carbon mineralization is a general measure of microbial activity and is the basis for decomposition. Nitrogen mineralization is an important piece of the nitrogen cycle and enables plant-available nitrogen to be released into the soil system. Both C and N mineralization are generally thought to increase immediately following harvest disturbance due to an influx of organic matter stimulating decomposition and microbial activity. However, it is unclear whether or not this state persists long-term. Extracellular enzymes play an important role in the degradation of compounds and cycling of nutrients by microbes in soil. In contrast to mineralization, which are direct measures of microbial activity, enzyme potentials provide insight into the capacity of the soil microbial community to perform processes related to decomposition and nutrient cycling. Previous studies have found conflicting results of enzyme potential following harvesting, and long-term studies are also limited. For our laboratory analysis, we measured total C and N by dry combustion. We measured active carbon by oxidation with potassium permanganate. To look at some microbial activity indicators, we set up a 28-day soil incubation, which measured C mineralization, net N mineralization, and nitrification over the course of that period. C mineralization was measured as soil respiration, and N mineralization was measured as the change in both nitrate and ammonium. 2 extracellular enzyme potentials were also measured. The first, beta-glucosidase, which from here on out I will refer to as BG, is a hydrolytic enzyme involved in cellulose degradation and often used for microbial acquisition of C. And N-acetylglucosaminidase, which I'll refer to as NAG, is also a hydrolytic enzyme, but it is involved in titin degradation. So now let's look at some data. Here we have the plots of total soil carbon and nitrogen. We see that each LTSP site has its own column. On the left, we have the core sites, starting with Council, followed by California and Umpqua. And on the right side, we have the affiliated with Matlock, Moara, and Fall River. On the y-axis, along the top row, we have total soil carbon. And on the bottom, we have total soil nitrogen. Both of these are in gram carbon or nitrogen per kilogram of dry soil. And along the x-axis, we have treatments. The core sites have compaction treatments, low and high, which are located within the shaded rectangle of each plot. And the organic matter removal treatments, OM0, OM1, and OM2, are in the non-shaded rectangle. You'll notice that the total N at Umpqua looks a bit unique with the entire plot being shaded. This is the plot that we will see when there is a simultaneous interaction effect of compaction and organic matter removal. So we'll have the filled circles representing different levels of organic matter removal within low compaction plots, and open circles representing different levels of organic matter removal within high compaction plots. The affiliated sites do not have compaction treatments, and so therefore they lack shaded areas. An asterisk indicates a significant difference between low and high compaction of that soil measurement, and upper and lowercase letters indicate a significant difference between levels of organic matter removal. Lastly, the horizontal line on each plot represents the mean of the uncut reference stands at a specific location and can be considered as a reference unharvested baseline. Within the results of total soil carbon and nitrogen, we observed a significant effect of organic matter removal at the core LTSP sites, where total carbon and nitrogen tended to decrease with increasing organic matter removal. We did not see a significant effect of organic matter removal at the affiliated sites, although Fall River did show a similar trend. At both California and Umpqua, we saw an apparent benefit of compaction, where highly compacted plots had greater carbon and nitrogen than low compaction plots. This may be attributed to having carbon and nitrogen containing organic matter delivered to the soil via mixing when the compaction treatments were applied, leading to increased soil C and N, or it may also be related to a sustained alteration of the soil environment, decreasing the capacity for decomposition, and in turn increasing soil storage of carbon and nitrogen. One pervasive effect of management on total carbon and nitrogen was the response in most severe level of organic matter removal, where the whole tree was harvested and the forest floor was removed. Compared to all other treatments, the OM2 plots consistently had the lowest soil carbon and nitrogen. Additionally, OM2 plots never recovered to that of the reference baseline, despite having greater than 15 years passed since that initial harvest disturbance. In contrast to total carbon, the response of active carbon to treatments had more variability within and between sites. There was a significant effect of organic matter removal at Umpqua and Molalla, however oppositely so, with a decrease in active carbon with increased organic matter removal at Umpqua, and an increase in active carbon with increasing organic matter removal at Molalla. At Council, we saw an interaction effect, with active C responding differently to organic matter removal treatments depending on the level of compaction. However, at the other core sites, there was no effect of compaction alone. Both low and high compaction plots had similar active C responses, which was also undifferentiated from the reference baseline. Overall, it was difficult to find common trends within the active C response. Although some previous research has suggested active C and total C are well correlated, we did not find active C to trend similarly to total C, at least consistently across sites. This may indicate that unlike total carbon and nitrogen, active carbon is more sensitive to individual site characteristics, precluding universal management effects. Here we have the graphs of soil carbon and nitrogen mineralization, with along the top row the y-axis being C mineralization, and along the bottom row having N mineralization. Both of these are in milligrams C or N per kilogram of dry soil. For C mineralization, we saw a significant effect of organic matter removal at California, Umpqua, and Molalla, where C mineralization tended to decrease with increasing organic matter removal. We again saw an interaction effect at Council, where varying levels of organic matter removal responded differently depending upon the compaction level. There was a significant effect of organic matter removal on N mineralization at Umpqua in California, where less N was mineralized in plots with greater organic matter removal. and there was also a significant effect of organic matter removal at Fall River, although it responded oppositely to the core site response. This may be attributed to Fall River being a highly productive site and management being less likely to have an apparent effect. We also observed a significant effect of compaction at California and Umpqua, although N mineralization responded oppositely at each site, with decreased N mineralized in high compaction plots at California and increased N mineralization in high compacted plots at Umpqua. Although the trends of C and N mineralization were inconsistent among treatments and sites, in all but one instance, OM2 plots had C and N mineralization less than the reference baseline, indicating a lack of recovery despite greater than 15 years having passed. Decreased mineralization capacity, as we observed within most OM2 plots, may be indicative of these severely harvested plots experiencing some microbial C or N limitation. And the last plots we'll look at today are enzyme potential activity. Along the top row, we have BG potential activity, and along the bottom, we have NAG potential activity. Both of these measurements are in micromole per gram carbon per hour. At the core sites, BG potential activity increased with increasing organic matter removal, and this was especially pronounced in the OM2 plots, where BG potential was consistently greater than that of the reference baseline. NAG activity was inconsistent across sites. Malala and Matlock had similar activity between organic matter removal treatments, although both were higher than that reference, while activity at Council, Umpqua, and Fall River was similar in all treatments, including the reference baseline. There was no effective compaction at any of the core sites in activity level of BG or NAG. Increased enzyme potential, as we saw with BG in those severe organic matter removal plots, may be indicative of increased microbial energy being put towards the acquisition of C, potentially indicating microbes in these plots are C limited to some extent. In terms of the response to nutrient pool soil properties, after greater than 15 years post-harvest, it appears that total C and N respond more universally to treatments than active C. Additionally, there is indication that greater than 15 years is sufficient time for recovery of soil C and N pools, except in treatment plots where management is severe, such as OM2. In terms of microbial activity indicators, our data suggests that after greater than 15 years, the responses of microbial indicators may be mediated by distinct site characteristics. That being said, severe management, such as OM2, exhibits some signs of having a microbial environment less conducive to normal function. Now, as we return to our original hypotheses, we found that our first hypothesis was supported by declines in soil C and N pools, regardless of prevailing inherent soil properties, and to a lesser degree in C and N mineralization within OM2 plots. These data exhibit what appears to be a compounding effect of pre-existing site nutrient limitations and decreased C and N pools, resulting in exacerbated microbial nutrient limitations. The OM2 response of lower mineralization capacity, coupled with increased potential enzyme activity across the core LTSP sites, could indicate a long-lasting impact of forest floor removal treatments on the microbial community and their important functions. Our second hypothesis was also supported, as we saw fewer effects of compaction compared to organic matter removal, and they were often even positive, such as the total C and N response at California and Umpqua. In this case, compaction may have aided in physically delivering soil organic material to the mineral soil following harvest, or may have created an environment less conducive to decomposition, therefore allowing increased C and N storage. And lastly, our third hypothesis was also supported. The site-specific responses of microbial indicators, including C and N mineralization and enzyme potential activity, indicate that dynamic soil properties did not respond the same across sites after 15 to 25 years. These indicators may be more dependent on unique site characteristics that moderate conditions for microbial activity. Overall, we can conclude that although often sites behave differently depending on inherent soil properties, the forest floor holds an important role in maintaining ecosystem services. By cultivating management practices that promote and protect this resource from degradation and that also take distinct site characteristics into consideration, we can improve our ability to sustain the health of managed forests and ensure continued productivity. Thank you. Oh,
Right,
and we have one more question I forgot to drag. So research suggests that there is no lasting effects of soil compaction on dynamic soil properties after what period of time? 5 to 15 years, 15 to 25 years, 25 to 35, or greater than 35.
Everybody take a crack at answering that poll question, and what did they find in this research, and maybe a chance to review what was presented. All right, well, the numbers seem to have settled down, so I'm going to end the poll and broadcast the results. Jordy, you want to go over the answers?
Yeah, you guys did great with 42% saying 15 to 25 years, which was the span of the ages of the LTSP locations that we studied. So good job.
All right, thank you, Jordy. And I just invite people to put their questions in the Q&A pod, and we'll take those as they come in. And Jordy, we do have some questions that have come in, and Sky asks, what do you think your research results mean for use of this method in general soil health assessments, especially forest soils, rather than cropland soils?
I think that my suggestion is that in general soil health assessments, using measurements that may vary less by distinct site characteristics would be important. I think this sort of leads into our findings with active carbon, which I think is used pretty frequently in cropland soils, and we did not find it to be that consistent across sites in terms of treatments in forest soils. And so I think that it's important to consider the different ecosystems and how there may not be one set of measurements that works for all.
All right, Darren has asked in the chat, what about the phosphorus as a nutrient pool?
I think that phosphorus is an important nutrient pool, but we did not look into it in this study.
I would just add that in forests of the Pacific Northwest, it's pretty rare to ever see a phosphorus limitation or a response to phosphorus, and so that was one of the reasons why we didn't focus on it. Other areas, such as the Southeast, phosphorus often tends to be a bigger limitation to forest productivity.
All right, another question in the Q&A from Megan. Great presentation. It appeared in your graph that potentially droughty sites had stronger responses to treatment. Were you able to compare response rates against edaphic variables?
No, but that would be an excellent thing to be able to do. I think that that would be a cool next step, would be to use some of our results and try and get at what of those distinct site characteristics might be causing differences or differential responses to treatments.
I'd
It's actually
also
very similar to a question I was going to ask about, would you speculate if there was any effect on like water tables, the presence of a water table? And did any of your sites have a water table? I don't know.
So I'll take a crack at that and also talk a little bit. Yeah, the different climatic regimes, I guess the challenge with this study is that in addition to that variation in droughtiness, if you will, there are also variations in soil texture, and so being able to tease those apart with 6 sites is a little bit challenging. With the exception of Council, the topography of the other sites tended to be relatively gentle or flat. I suspect that there were, at all these sites, there's going to be a water table at some depth. I don't think it was ever shallow enough to be necessarily a real concern, if that was kind of where you're going at, Sean?
Another question in the chat, was there any difference in horizonation observed, like thickness of O horizon?
I think just from observation as we were sampling these sites, there was a little bit of difference. Some, I would say, treatment and definitely between sites, but for our study, we just removed the O horizon and just sampled the mineral soil.
Right, well, we seem to have exhausted the questions. So I want to thank you, Jordy, and thank you, Dave, for your time and effort to make this presentation. And thanks to all the participants for joining in. We had more than 70 people join today's webinar. The on-demand recording of this webinar will be available on our YouTube channel, along with over 150 webinars and training videos. Simply search for NRCS, NSSC, or this topic in YouTube, and you'll find the recordings. Please feel free to let your colleagues within and outside of NRCS know about this training opportunity. And this concludes our webinar presentation.